the bad news first. Bad news is that I have a post-independence gift for you guys, which is called test number 1. That will be Thursday, or day right after Independence Day. So hopefully you guys would have recovered from your Independence High. And as usual, the test will open from 4 p.m. and close at 11 p.m. Is that okay, Cheyenne? The time with you?
Yes, sir, that work.
Okay, I don't come back from church, right?
Yes, sir, I done finish prayer.
Okay, that's
Let
good.
pass.
Yeah, all right. It's good. The good news is that we won't have any class Thursday morning, so you'll have time to recover from whatever high you had or your goma, and you'll have time to recuperate and freshen up for the afternoon and the night for the test. So we won't have classes Thursday morning. Is that clear? All right, so no classes Thursday morning. You guys will be recovering and get ready for your tests in the afternoon and the night. Okay, any questions? All right, so no questions. Let's continue our discussion. Be reminded, I mentioned already what will be on test number 1, right? I mentioned at last class. So take note that what we're discussing today will not be on the tests. Okay. Last class we mentioned Morgan. So Thomas Hunt Morgan, he was a geneticist, evolutionary biologist, and he did a lot of follow-up experiments to what Mendel did. And so I mentioned about cross he did with purple flowers and long pollen grain, with red flowers and short pollen grain. And based on the results of the F2 generation, he did some calculations and he came up with the significance of those calculations. All right. We will go over that in just a few. But Morgan, besides working with Garden P and those flower color and so on, Morgan also did some experiments with Drosophila, which is a fruit fly. All right. So let's look at Morgan's linkage experiments with Drosophila. Remember, this all has to do with linkage and partial linkage and so on. So we said that partial linkage is closely related to the crossing over that takes place in prophase 1, right? So the experiment that Morgan did, him and his students, when it comes to fruit fly, is that Morgan crossed 2 different flies. Here you have the female fly that had white eyes and small wings. And he crossed that with a male fly that had big wings but red eyes. So take note of the phenotypes of the parents from the P generation. Take note. A female fly with small wings and white eyes crossed with a male fly with large wings and red eye. Okay? The F1 generation had phenotypes whereby you had some females, as seen here, some females that had big wings and red eye. Take note of this term, wild type. Wild type. The term wild type means the normal fly. So that is what the normal fly looks like. So the normal fruit fly has big wings with red eyes. And so that female fly from the P generation that has small wings with white eyes, that is a variant of the wild type. Is that understood? So when you hear the term wild type, it is referring to the normal organism. And any variation from that would be a variant. So in the F1 generation, they obtain some females with the wild type phenotype, so large wings with red eyes. But they also got some males that had small wings with white eyes. So Morgan and his students decide, let's obtain the F2 generation. And you do that by crossing two members from the F1. And so they observe from the F2 generation, something happened. Something happened in the F2 generation, and we'll observe that in just a few. So here you have flies that have small wings with white eyes. So those resemble one from the P generation, small wings and white eyes. And then they also obtain wild type, which means big wings with red eyes. Those also resemble one from the P generation. So those are called parental phenotypes, all right? And the total number of parental phenotypes obtained were 1,541. So take note, small wings with white eyes resemble the female from the P generation, while big wings with red eyes resemble the male from the P generation. But Morgan and his students also observed something whereby they observed some flies that had big wings with white eyes, which means that they combine traits from two parents. So big wings with white eyes. And then they also observed some that had small wings with red eyes. Those also are combining traits from two of the parents. And so these ones are referred to as recombinant phenotypes. All right? So those that combine traits from the two parents. All right, so whether it be small wings with red eyes or large wings with white eyes, those are called recombinant phenotypes because they combine traits from two of the original parents. And so they obtain 900 recombinant phenotypes. So by adding up the 1,541 parental phenotype and the 900 recombinant phenotypes, they obtain a total of 2,441 total flies. All right. So by finding the percent of recombinants, they will divide the 900, all right, over the total times 100, and they get 36.9. If you recall from the last class, that number has significance, and it has two significance. Anybody can recall what does that mean, 39.9? What's the significance of that number? Anybody recall? Nobody recall? At 30, .9 two genes. Genes that is responsible for the size of the wings, and the gene that is responsible for the color of the eyes. 6 point. So those two genes are in frequency of crossing over that took place in prophase 1. All right. So it says, to explain the recombinance, Morgan proposed that in 36.9% of the meiosis, an exchange of genes had occurred between the two X chromosomes of the F1 females. And since males are hemizygous, then no genetic exchange occurs between the non-homologous X and Y chromosome. All right. So 36.9% of the meiosis, there was an exchange. So there was 36.9% crossing over that took place in prophase 1. In all cases, the parental phenotypic classes were the most frequent, while the recombinant classes occurred much less frequently. Morgan's general conclusion was that during segregation of alleles at meiosis, certain ones tend to remain together because they lie near each other on the same chromosome. And usually when you have genes right next to each other, it means that they are inherited together. They are passed on together, all right? We will look at mapping genes and determining gene order in probably just a few. So gene recombination. In the 1930s, Crichton and McClintock did experiments with corn, and the process of a chromosome segment breaking off from one chromosome and reattaching to another is called translocation. So trans has to do with a change and a change in location, all right? Cytologically distinguishable features such as these are called cytological markers, and the genes involved are called genetic markers or gene markers. So all those genes that are on that chromosome segment that breaks off and reattaches itself to a different chromosome, all those genes in that segment are called genetic markers or gene markers. Locating genes on chromosomes, mapping techniques. Crossing over takes place in prophase 1 of meiosis and involves only 2 of the 4 chromatids for a given crossing over event. Genetic experiments can be used to determine the relative position of genes on chromosomes in eukaryotic organisms. And this process is called genetic mapping. Genetic mapping is the creation of a genetic map, or it is called a linkage map, where it shows a chromosome and then the position of the different genes on that chromosome. All right, so we talk about crossing over taking place. Remember, we said that this occurs in prophase 1, and it's a case whereby right after synapsis, you have the exchange of genetic material from these homologous chromosomes. 2 different ways of mapping are distinguished. So you have genetic mapping uses classical genetic techniques. All right, for example, pedigree analysis or breeding experiments, and this is used to determine sequence features within a genome. Test cross, for example. We discussed test cross in the past, and we know that test cross is a cross between an unknown genotype crossed with a homozygous recessive. And the purpose of that test cross is to determine the genotype of that unknown. And test cross is better because the distribution of phenotypes is a result of segregation events in only one of the parents. the other parent contributes only recessive alleles to the progeny, right? So here we have, remember, I said that test cross is always, you always use a homozygous recessive in a test cross. And this homozygous recessive will only be contributing recessive alleles to that progeny, all right? And so based on the results of that test cross, you'll be able to determine the genotype of the unknown parent. Then you have modern molecular biology techniques, and this is used for the same purpose, and it is usually referred to as physical mapping. Loci or locus is the position of a gene on a genetic map. So Morgan thought that the characteristic crossover frequencies for linked genes, remember what are linked genes? All those genes that are on the same chromosome. So the crossover frequencies for linked genes might be related to the physical distance separating the genes on the chromosomes. Morgan's student suggested that the percentage of recombinants produced by crossovers in gamete production could be used as a quantitative measure of the genetic distance between two gene pairs on a genetic map. So this distance is measured in map units, all right, and it is abbreviated MU. So this is the unit used for distance of genes on a chromosome. So a crossover frequency of 1% between two genes equals 1 map unit, or 1 centimorgan, small c, big M. So these are the two units used to measure distance between of genes on a chromosome. So map units or centimorgans. So when you get a crossover frequency of 5%, that is indicating that these two genes that you studied are 5 map units apart from each other on the chromosome. So the crossover frequency is equal to the distance between the two genes on the chromosome. Is that understood? All right, so basically what Morgan and his students did was crossing these flies and then obtaining the F2 generation, and based on the results of that F2 generation, they calculated how many were recombinant phenotypes, and based on that find that percentage, which is equivalent to the frequency of the crossing over events in prophase 1 as well as the distance of these two genes on that chromosome. Similarly, what I explained last class, all right, whereby they use sweet pea, right, purple flower and long pollen grain crossed with red flower and short pollen grain, okay? All right, let's move into bacterial genetics, mapping genes in bacteria. One thing we should note is that bacteria are prokaryotes, and so when it comes to the genetic material, we're talking about one single circular chromosomal DNA, all right? So the genetic material for prokaryotes is much more simple than eukaryotes, right? So we're talking about one chromosome here, while in eukaryotes, for example, take human beings, we're talking about 46 chromosomes, all right? Where it gets a little bit more complicated in terms of mapping genes. E. coli is an extensively used bacterium in genetics and molecular analysis because it can be grown in a simple defined medium and can be handled with simple microbiological techniques. E. coli is found most commonly in the large intestines of most animals, including humans. Bacterial cells are relatively small compared with eukaryotic cells. Its cytoplasm is full of ribosomes, while the central nucleoid region contains the genetic material, which is a single circular chromosome consisting of DNA, and there is no nuclear membrane. Genetic material can be transferred between bacteria by three main processes. 1, transformation, 2, conjugation, and 3, transduction. All right? So take note. In prokaryotes and in bacteria, remember that there is no sexual reproduction. So the way how you can have genetic mixing is through these three ways: transformation, conjugation, and transduction. In all organisms that reproduce sexually, when you get that sperm from the male and it unites with the egg from the female, you have genetic combination. Genes are combining, right? But in these bacteria that produce primarily by binary fission, all right, you don't have that taking place. It does not mean that genetic material cannot be exchanged and you can have transformants. You can have a mix of genetic material through these three methods: transformation, conjugation, and transduction. In all three cases, transfer is unidirectional, whereby you have a genetic material moving from one organism going over to the other organism. All right? There is no true diploid zygote because fertilization does not exist. So there is no fertilization or syngamy, so you don't obtain a true diploid zygote. It's just a transfer of genetic material from one organism to the next. Only genes included in the circular chromosome will be stably inherited. All right. So let me ask you, in this particular case, when we're talking about transfer of genetic material between bacteria, would this be considered vertical gene transfer or horizontal gene transfer? So the transfer of genetic material between bacteria using any one of these three: transformation, conjugation, transduction, would that be considered vertical gene transfer or horizontal gene transfer? Anybody?
Horizontal.
Horizontal. All right. So that would be considered horizontal gene transfer. So bear in mind that vertical gene transfer, for example, occurs in sexually reproducing organisms, for example, human beings, whereby genes are passed from generation to generation to generation. That would be vertical gene transfer. But in this particular case, we would be talking about horizontal gene transfer. Got that? So for these reasons, genetic transfer processes are different from those in eukaryotes, because most eukaryotes, the gene transfer would be primarily through sexual means, fertilization, okay? Let's look at each one of these and understand the concept and how they differ from each other. So transformation, right? Bacterial transformation. This involves DNA from a donor strain, and that is extracted and purified. The extraction process breaks the DNA into small, linear, double-stranded fragments, and this genetic material is then added to a suspension of recipient bacteria with a different genotype. All right, so some key information here is that you have a donor strain, which means that it comes from a different bacterium. The only way how you can successfully transform bacteria is that if you have two different genotypes. So you get a donor strain, you extract the DNA from that donor strain and you purify it, and then that DNA is broken up into smaller fragments, and that is mixed with recipient bacterial cells in suspension of a different genotype. So the recipients whose phenotypes are changed by a recombination event are called transformants. So if there is any bacterial cell that took up those small DNA fragments from that donor strain and got that DNA incorporated into its chromosomal DNA, those are called transformants. So any bacterial strain can be the donor strain and any strain can be the recipient. Only if there are genetic differences between donors and recipients will transformants be obtained. All right? So that is extremely important when it comes to transformation. You have to make sure that you have two organisms that are of different genotype. So you get the DNA from one organism, you extract that, you purify it, cut up the DNA, and then you mix it with the recipient cells in a tube. And then you hope that those DNA fragments would be absorbed and integrated into the chromosomal DNA of the recipient cells. And that is how you'd obtain a transformant. All right. So we have the diagram here, and if we focus our attention on the left side first, whereby it says transformation with DNA fragments. All right, A. All right. So here you have DNA fragments. Remember, these were taken from a donor organism. It was extracted, purified, and broken up into smaller fragments with the use of restriction enzymes. And here you have this bacterial cell that would represent the recipient. So these are co-cultured, all right? So these are cultured together. And then you have uptake of DNA, whereby these fragments get inside that recipient cell. And once it gets inside, a couple of things can happen. If you have integration of this portion of foreign DNA into the chromosomal DNA, like what you see here, then that would be a stable transformation. That would be a transformant. That cell would be a transformant. However, there are times when before that DNA fragment is able to integrate itself into the chromosomal DNA, it got degraded, all right, by different enzymes. So you can have some enzymes that actually degrade the DNA. And if that happens and it does not integrate itself into the chromosomal DNA, then that would be an unsuccessful transformation event. All right, so for you to obtain a transformant, the foreign DNA must integrate itself stably into the chromosomal DNA of the recipient cell, okay? Now you can also have transformation with a plasmid. All right, so recall, what are plasmids? Plasmids are these small circular DNA molecule, much smaller than the chromosomal DNA, and they're found floating in the cytoplasm of these prokaryotes. All right, so you have many different plasmids. But you can get a plasmid from a donor cell, and you co-culture that plasmid along with the recipient cell, and then you can have uptake. All right, so that plasmid finds its way into the donor cell, and once it is inside the cell, then that is considered a stable transformation. Why? Because there is where plasmids usually reside. Plasmids do not integrate themselves into the chromosomal DNA of any prokaryote. All right, so plasmids exist independently of the chromosomal DNA of that prokaryote. They are not integrated into the chromosomal DNA. So once that plasmid gets inserted into the recipient cell, that will be considered a stable transformation event, and that those cells will be considered as transformants. Is that understood? All right, so here we have transformation whereby you have, this would be the donor cell. So it is from this donor cell whereby you would extract the DNA from here, purify it, and then it is cut up into pieces, and that is then mixed with the recipient cell, and whereby you'll have fragments of that DNA enter the recipient cell and integrate itself into the chromosomal DNA of that particular recipient cell. So you end up with a transformant, all right? So transformant because it has a recombinant DNA. Recombinant DNA means that it has portions of DNA from two different organisms. All right, so you have from the donor and the recipient. Okay, so that is transformation. Some bacteria, such as Bacillus subtilis, can be easily transformed in a test tube. Wild type E. coli. What does wild type mean? Anybody recall? The normal one, right? The one that did not suffer any change. So the wild type organism is a normal organism. If there are any changes to the wild type, then that would be considered a variant or a mutant because there are changes. So wild type E. coli, however, is not readily transformable because the bacterium produces enzymes that rapidly degrade incoming DNA. Okay? Cells are treated chemically to make the membrane more permeable to DNA. So you sort of open up pores to allow those foreign DNA to come through the pores, all right, and come inside the cell and integrate itself into the chromosomal DNA. That process of creating these pores on the membrane, that is reversible. So as soon as everything is done, it returns back to its normal state. Those cells that are able to be treated and make that membrane more permeable, those cells are called competent cells. In terms of mechanism, there are two types of bacterial transformation. The first one is natural transformation, in which bacteria are naturally able to take up DNA and be genetically transformed by it. So natural transformation. So bacteria are naturally able to take up DNA and be genetically transformed by it, such as the case of Bacillus subtilis. And then you have engineered transformation, in which bacteria have been genetically altered to enable them to take up and be genetically transformed by added DNA, such as the case of E. coli. All right? Using transformation, it is possible to determine gene linkage, gene order, and map distance. If co-transformation occurs at a frequency that is substantially higher than the product of the two single gene transformation, then the two genes must be close together. Let me try and explain this. Co-transformation means that whenever you're going to get a piece of DNA moving from a donor cell over to a recipient cell, co-transformation means that that is not one gene that went there, it's more than one gene. So when you have co-transformation taking place between two genes, that is an indication that these two genes are right next to each other on that chromosome. I'll try and repeat that so you can understand, because I will have you guys determine gene order on a chromosome based on co-transformation data. I'll repeat. If co-transformation occurs between, if I tell you that co-transformation occurs between genes X and Y, So remember, we're talking about a bacterial cell that on that bacterial chromosome, you have a portion that was transferred from the donor cell. And if we observe that co-transformation occurs between X and Y, then it simply means that X and Y are right next to each other on that chromosome. Is that clear? I will repeat it one more time. My co-transformation data, and if I say that co-transformation occurs between two genes, A and B, all right, which means that both A and B were transferred from that donor cell over to the recipient cell. It's an indication that these two genes are right next to each other on that chromosome. Okay? So a gene order can be determined from co-transformation data. All right? So if I say that co-transformation occurred between X and Y, all right, and co-transformation also occurred between Y and Z, then we know that the order will be X, Y, Z. All right? But co-transformation does not occur between X and Z, which is an indication that they are not next to each other on the chromosome. Okay? So I will put up on the screen some co-transformation data, and you can determine the order of these genes on the chromosome. It says write the gene order on a chromosome if co-transformation occurs between genes R and O, C and A, S and T, R and T, A and S, but never between S and C, T and A, R and S, T and O. So go ahead and write the gene order, all right, the order of these genes on the chromosome, and when you get it, you can write it in the chat. I'm feeling good today. Today is pre-independence. It's Independence Eve. So for 5 points. Write the gene order for me. Who write it first in the chat? Okay, so Marva Ramirez has gotten her 5 points, which will go towards the next quiz.
Thank you, sir.
Do you want it towards the next quiz or towards test number 1?
Towards test number 1, please.
Test number 1, test coming up on Thursday. Okay.
Yes, sir.
Five points for Marva. So, you care to explain to the class, Marva, how you came up with Castro?
Sir, I just kind of move around the letters, but I first write out the list, R and O to C and A, and S and T, R and T, E and S, and I remove some of the extra, one or two of the extra letters. And then my following step, I went to that said they never had any connection between the S and T, T and A, R and S, T and O. I move on the letters again, sir.
Okay. Recall that piece of information that I repeated about 4 times that says if co-transformation occurs between 2 genes, it's an indication that these genes are right next to each other on that chromosome. So when I tell you co-transformation occurs between genes R and O, it means that R and O are right next to each other on the chromosome. R and O. So you write that down. Those are 2 genes that are right next to each other on that chromosome. Okay? And then it says C and A, co-transformation occurs between C and A. S and T, as well as R and T. So if co-transformation also occurs between R and T, then it is telling me that T is next to R. So I can safely write T here. But it had said that co-transformation also occurs between S and T, which means that S is next to T, so I can write S here. And then it says co-transformation occurs between A and S, which means that A is right next to S, so I can write A here. And then it says between C and A, which means that C is next to A. And that is how Marva came up with Castro, which is the order of these genes on that chromosome. And we can corroborate it by saying that co-transformation never occurs between S and C. See, are C and S next to each other? No. Never between T and A. And never between R and S. And never between T and O. All right, you all got that? So you can determine the order of genes on a chromosome based on co-transformation data. All right, so always think about transformation, whereby you get DNA from the donor, is extracted, purified, cut up, it's co-cultured with recipient cells. And when a piece of fragment of that DNA moves over to the recipient cell, you can have several genes moving over at the same time. All right, and based on these co-transformation data, you are able to determine the order of the genes on the chromosome. All right, so I have one more for you guys. It says, write the gene order of a chromosome if co-transformation occurs between genes E and L, N and Y, D and E, Y and L, D and E, but never between E and A, D and L, N and E, for 5 points. write it in the chat. Marva can do it, but Marva don't get 5 points already, so it will have to be somebody else. Is that how you pronounce it? Ziani? Ziani Moore?
Yes, sir.
Okay. Ziani has 5 points towards test number 1. Even though you write it the other way around, like what Madison wrote there, it would still be correct. All right? Like what Madison wrote would still be correct. She just write it from the tail end. All right? But Ziani got it correct. Adeline. So that would be the order of these genes on the chromosome based on co-transformation data. All right? So, well, you had it, but you didn't write it, Lourdes. The idea was to write it in the chat. Who write it first in the chat? Delvin said probably the man typed too slow. All right. Anyway, Ziani Moore got her 5 points towards her test. Anyway, guys, we're gonna stop here for today, and then when we resume class next week, Tuesday, we will continue talking about the other 2 forms of how genetic material can be transformed between bacterial cells. So remember we said transformation, which we looked at here. We can transfer DNA fragments, or we can also transfer plasmids into bacterial cells. Next time we meet, we will look at conjugation, and then we will also look at transduction. These are 2 other ways how genetic material can be passed from one bacterial cell to the next. Okay? Any questions before we leave? All right, so guys, take care. Enjoy Independence Eve. Enjoy Independence Day. Have fun. And then I wish you all the best on your tests on Thursday. All right, so take care.